So in this video I'm going to show you how to make a wet mount. Wet mounts are useful when you want to look at live cells or a very tiny organism. So for example, you might make a wet mount when you're going to look at a plant leaf from a water plant and try to see chloroplasts and maybe even some cyclosis, which is when the chloroplasts are streaming around inside the live cell. You may also gently scrape out a cheek cell or a couple of cheek cells from your mouth and then put them onto a glass slide like this. And you might even want to stain them using methylene blue so they show up better. Another application in this lab is that we start off the semester looking at vinegar eels. And so they're not eels at all, they're little worms, roundworms. And they swim around, and sometimes they can be hard to actually chase after them. So in that case, when you're making your wet mount, you may add a thick, viscous solution called Detain that makes it hard for the worms to get through. In other words, it slows them down and makes it easier for you to view them. But in either case, we're going to put a specimen and some fluid onto a regular glass slide. So the first thing you want to do is make sure that the glass slide is clean, so there's not a bunch of smears and stuff that just kind of makes it harder to view your specimen. And then the next thing is, as you can tell, it sounds like we're using some fluids. Well, we want to make sure we don't get that fluid onto the objectives of our nice compound microscopes. So for that, you would need a cover slip. And so starting off with these glass slides, they usually come in big boxes like this, and then those, when you're done, you should wash them off just like you wash your dishes in the kitchen and put them in a drying rack over by the sink. When it comes to these little cover slips, which is the box that's open here, these are we don't wash. We just throw them out into a dedicated container. Usually it's a beaker over by the sink, and then you just put them in there and we dispose of them. All right, so glass slide we want to wash and save. Cover slip we want to throw out in our special container. Okay, so in this video I'm going to just make a wet mount out of a leaf specimen. And so in this case I'm going to take this leaf and I'm going to put it onto the glass slide. So the next is that, well, I don't need to detain because it's not moving, but I want to put a small drop, maybe two on this case. In general, I would start with one because you do not want so much fluid that your cover slip kind of starts floating away, and you don't want any fluid outside of the cover slip. So then I take my cover slip and I want to try to get, I don't want to trap air bubbles underneath. And so a good technique is to angle it at a 45 degree and slide it up, get a little capillary action, and then drop it. And that looks pretty good. So now I'm looking around. There is no excess water. If there is excess water outside, you need to go get a well-absorbing paper towel and kind of blot on the side to remove that. Remember, this cover slip is here to protect the objectives of the compound microscope, and so obviously if you're having a lot of stain or water here, there's a chance that you would actually contaminate that objective. So please make sure that there's not excess water. If it is, you need to remove that, and then at this point we're ready to slide this in to our compound microscope and view it as with our normal technique as we usually do. All right.